Hello, my name is Taylor.Tibbs, and I am a biology major, German minor here at Carthage College. I am class of 2017, so I'm a junior this year, and I have been nominated for two internships positions, one being a Purdue University research experience that I had last summer, and another one being a research opportunity that I performed here at Kenosha, currently in the Kenosha Parks Department. So, a little bit about myself. I have spent the past 3 years of my life being really focused in research. It's a passion of mine that I have started since I was a freshman. And during my freshman year, I was a part of the Phage Hunters program here at Carthage. So this is a year-long coursework study that is funded by the Howard Hughes Medical Institute, in which we discover and isolate bacteriophage from the environment, and we have their genome sequenced, and we annotate that genome, and we actually get a publication on PubMed. And it was a really great experience and really shaped the career path that I have developed now, which is to go to grad school and study infectious disease and microbiology, and potentially even earn a PhD, MD, to apply the clinical applications of microbiology as well. I began an independent research study with a few other biology students here at Carthage studying rotococcal phages, which is another bacteriophage that infects the bacterium Rhodococcus erythropolis. From there, I then went on to my summer internship at Purdue University in the biochemistry department. From there, I started my thesis work working with a genomic editing tool called CRISPR-Cas, and I would like to use it to edit bacteriophage DNA of lambda phage in vivo. And so this would be a novel project that has not yet been done, and I'm really excited to work on it. This summer, I am also participating in a broad internship for 3 months in Greifswald, Germany, studying Staphylococcus aureus and various mutant phenotypes there, and I'll learn a ton of more exciting techniques. So I have really gotten on the research bandwagon, and I'm excited to continue. I'd like to talk about my Purdue research internship first, and then I will get into a little bit more about the Kenosha Parks internship. And so, at the Purdue University, I was in a biochemistry laboratory, and I was studying a ribozyme called DRZ-MTGN1, which had an atypical catalytic trend, and we were wanting to see if this was due to different conformations at varying levels of magnesium ion concentration. And to spare all the great details, I was able to sort of develop my own experimental design with a graduate student in that laboratory as far as step-by-step process of how we were going to accomplish this task to get our goal met. And we also had a lot of problem solving for when those experiments didn't go quite as planned. So I also learned various different techniques in biochemistry, and also to use many different advanced equipment there at Purdue University, and it was a real treat and an amazing opportunity. And so those techniques that I have acquired and equipment that I've been able to become familiar with are going to be vastly important for my graduate school applications, which makes me a more competitive applicant now that I've had those research experiences. From this experience, I actually was able to develop even more insights into crystallography. Before participating in this internship, I had taken a structural biology course here at Carthage that studied the molecular structure of proteins and how the crystallography techniques work. And it's very different to be sitting in a classroom setting being lectured about a technique and see pictures on a slide of how you would put this chemical with that chemical than when you're actually doing it in lab. And it was really eye-opening and allowed me to get a better feel for what science really is. What is it like to do that science in a graduate laboratory in a larger university with all these high-tech equipment. And it was invigorating, it was elating. I I had a blast, and I really want to go back and can't wait to do it again this summer. I was also able to disseminate my research from Purdue University at not only a conference at Purdue University itself, presenting to the department, but also at the Mid-States Consortium in St. Louis at Washington University. I gave a poster presentation there. I also gave an oral presentation, a 15-minute talk here at the National Conference of Undergraduate Research in the University of North Carolina, Asheville, and will continue to give a talk on this research at Celebration of Scholars this summer. So if you have time, feel free to stop by. The other internship I would like to make note of here is the Kenosha Parks Department internship that I'm participating with Matthew Collins, in which I am working on the local prairie restoration of an old agricultural field here. This project involves taking on tasks such as determining the native plant species that would be best suited for this area given its soil texture and temperature and various drainage of the soil and so on. So putting together a diverse species list to plant in this area, but also to take soil samples and have them tested for their nutrient levels to ensure that everything will be ready to go for this prairie restoration. So that involves actually getting out there into the field and picking up those samples and sticking a soil core into the ground and sticking the Ziploc bags and marking down their GPS coordinates. So there's a little bit of field work involved, but there's also an immense amount of background work that also has to be done as far as developing that species list and estimating the equipment costs of seeding and taking maintenance of this prairie. Our ultimate goal is to produce a grant proposal, so I have to find a grant source for this prairie restoration and be able to put it in a presentation format for a proposal so that way we can receive the money to do this prairie restoration. So this gave me an opportunity to experience the other side of science, and in a professional aspect, I spend the majority of my time in a lab studying molecular microbiology techniques, and this allowed me to experience the ecological side, the environmental focus side, and to actually get out there and be in the environment even with some of the tasks of the field. And so in a professional sense is also extremely valuable, as I was able to cultivate new relationships with members at the Kenosha Parks Department, but also honing my skills as a writer and a presenter and engaging in a business-like setting, in a formal business-like setting. Thank you for your attention.